Hi, in fifth day of biology class, today I'm going to present cell structure and function. Please remember that in my YouTube channel, there are different playlists where you need to look for your area of interest. For example, for biology tutorial, I have different playlist. And for bioinformatics, different playlist is available. Then for bioinformatics, computational biology, different playlists are available. So please subscribe the channel in order to get the update from time to time. So in this class, you will be able to learn the structure and function of the cells, how cells could be studied by using different techniques, and to learn the difference between prokaryotic and eukaryotic cells. Then there will be some more, but in another class. But today only I will be able to present the cells, how the cells are studied, the cell components, different components of the cells. So let's start from here. There are different ways to study, but one of the most common and the oldest one is microscopy. Of course, there are lots of advancement in microscope technology. So, but you just need to understand that microscopy is one of the main source to visualize the cells. And if you want to go further depth visualization, you need to have more and more advanced version of microscopes. And in general, the microscopes, how the microscope works, all these things, you could understand and learn from some other sources. Here I will be switching to the topic of our interest. So in general, the cell theory, if you want to understand. So before you go for cell theory, you can see these are the images of the cells visualized by using different technologies. And some definitions, how the cells could be defined, and living cells, dead cells, these are some far basic things. And I will not go for deep, but I will prefer to say that you just need to think the cell in terms of like the cell, which has nucleus, cell membrane, or if it is plant, it's a cell wall, and then some components of the cells which could maintain the cell's biological functions and activities. And there are different theories behind this, which I am not interested to go, because here you are to learn the main theme instead of learning the history of biology or the cell. So the prokaryotic and eukaryotic cells, in general, if you want to know what is a prokaryotic cell, you can say in terms of definition that it is single-celled, unicellular organism that lacks a nucleus or any other membrane-bound organelle. We will, like, there, as I said in the previous slide, that the cell has nucleus. So there are definitions which are varying. That is why there is a subsection in every book, the cell theory, something like this. So plant cells are slightly different. Animal cells are slightly different. Prokaryotics are different from eukaryotic cells. So if you want to understand a prokaryotic cell structure and function, generally it has different components. And in terms of component, you can say nucleoid, then plasmid, ribosomes, cytoplasm, fimbria, plasma membrane, cell wall, capsules, cell capsules, and flagellum. So here nucleoid region of cytoplasm where prokaryotic DNA is located, similar to nucleus, as the nucleus, the eukaryotic cells, they have it. Then plasmid, which is extra chromosomal double-stranded circular DNA, which encodes genes for fertility. Then it comes cytoplasm, gel-like matrix with water, enzymes, and cell structures. Then ribosomes. Of course, I will be discussing there are different types of ribosomes. Here, 70S ribosomes are present. That is a site for protein synthesis. Inclusion, storage reserves, and plasma membrane, which is made up of phospholipid bilayer and proteins. So if you want to see the same thing here, you can also see with some text description. For example, flagellum is used for motion. So basically this slide is giving you more idea in terms of picture. Instead of learning the text, you can understand what is the role of flagellum. Then it means it is for motion. Capsule reserved for food and protection. Then enfolding of cell surface membrane, which allow photosynthesis or carry out nitrogen fixation. Then plasmid, a small circle of DNA, pili for attachment to the other cells or surfaces. So, and it is also used or involved in sexual reproduction. Now, cell wall, similarly, protection and some other roles, then cell surface membrane, then cytoplasm, circular DNA, ribosomes. So this is the simplest version of living organism cell. Now, eukaryotes, like highly modified form of prokaryotes. This is what you can say, advanced version of prokaryotes. So eukaryotic cells, again, I'm saying I'm not going deep inside, but you can understand a eukaryotic cell has a membrane-bound nucleus, well-defined nucleus, and other membrane-bound compartments or sacs, which is called, they are called as organelles, that is cell organelles, which have a specialized biological functions. The, like, it's like if you want to go deep inside in studying each organelle, you will be embedded. But at class 9, 10 level or 11, 12th level, you need to know the basic level. So here, let's start from nucleus. Then nucleus has nuclear envelope, which is membrane enclosing nucleus. Protein lined pores allow material to move in and out from the cytoplasm to the nucleus, nucleus to the cytoplasm. And chromatin, which is like DNA and the associated proteins. So the chromatin is here, what you are saying. Then nucleolus, which is here, condensed region where ribosomes are synthesized or formed. Then peroxisomes metabolizes the paste. This is also very important, similar to lysosome, which digests like food and waste materials. Now, endoplasmic reticulum, there are two types of endoplasmic reticulum or ER. Rough endoplasmic reticulum, RER, smooth endoplasmic reticulum, SER. So, rough endoplasmic reticulum is associated with ribosomes and makes secretory and membrane proteins. Smooth endoplasmic reticulum makes lipid. Now, cytoplasmic microtubules form the mitotic spindle and maintain cell shape, which you can see all these things. Yeah. Then centrosome, microtubule organizing center, and that is here. Intermediate filaments, fibrous proteins that hold organelles into a specific location or place inside cytoplasm. Microfilaments, fibrous proteins forms the cellular cortex. And here you are seeing plasma membrane. This is here. Lysosomes digest food and waste materials. Then Golgi apparatus modifies proteins. Cytoplasm here. Mitochondria produces energy and then vacuum. So there are different cells and their sizes, known sizes have been mentioned here, which is you can see here from 0.1 nm to 1 millimeter. Now, what you can see from microscope and what you can see from light microscope, what you can see from naked eyes, this is clearly mentioned here. Now, prokaryotic cells versus eukaryotic cells in terms of a structure, this will give you much better insight to understand a structural difference, or in terms of diagram to understand the main differences between prokaryotes and eukaryotes, which I have already explained in the previous slides. But if you feel difficulties in understanding those 2 3 slides, you can directly look over this slide where you can clearly get a map in your mind the main difference between prokaryotes and eukaryotes, and that is here. Now, if you want to understand in terms of text, let's say comparison of prokaryotic and eukaryotic cells or organisms, examples, Monera, then eubacteria, archibacteria, protists, fungi, plants, animals. Levels of organization, that is single celled, levels of organization, single cell, and could be multiple cellular cells also. Then typical size of cell 1 to 10 microns, here 10 to 100 microns in eukaryotes. Cell wall is present in prokaryotes. Almost all have cell walls. And then here fungi and plants have, but it is cell wall is not present in animals. Organelles, here usually no organelles. Then here, as I have mentioned in eukaryotes, there are well-defined, well-known functional organelles. Metabolic activities, anaerobic and aerobic. And here in eukaryotes, it is mostly aerobic metabolism. Genetic material, single circular double-stranded DNA. Here, complex chromosomes, usually in pairs, each with a single double-stranded DNA molecule and associated proteins content in a nucleus. Mode of division, binary fusion mostly, and here mitosis and meiosis using a spindle followed by cytokinesis. Now, more deep, if you want to understand, it is DNA, which is naked DNA without histones. DNA is circular. Genes do not contain introns. DNA found in cytoplasm. In eukaryote, DNA associated with histones, that is not naked. DNA is linear. Genes may contain introns. DNA found in nucleus. Then internal structure.